Okay, so go ahead, Emma. And Emma's also mentioned that she's happy to be interrupted. So if you have a question, either just go ahead or put your hand up and I'll hopefully notice it.
Great. So yeah, no, it's a real pleasure to be invited to present for all of you today. I see this seminar series is a lot around kind of quantitative biology, and I assume that's sort of the core interest of a lot of people who are listening. So I've tried to make this presentation such that firstly, you don't need to be an immunologist to understand it, and secondly, to focus on more where we have used quantitative methods to try and understand the gut immune system. And I'm very interested both in getting some feedback on what you think, what could be developments of this, where you think this could potentially go, and also maybe whether there are new modeling methods and things that could play in, because this is, we're good immunologists in the group. We have some modeling expertise, but it's obviously not the thing that we do best. So to get everybody on the same page to start with, I have a nice explanation of the sort of challenge that we're working with the intestinal immune system. So you might have realized from my accent and the introduction about where I studied as an undergraduate that this is a house in London, which is not very far from where I grew up. And the people who live in this house look quite special, right? They have this tendency to wear lots of diamonds and bear fur and things that you don't normally see sort of down the shops on a normal day. And this is very useful if you're a security guard who works in Buckingham Palace, right? Because it's very easy to identify if a member of the public gets into Buckingham Palace. It's very clear that this is a problematic situation and you need to throw everything in the kitchen sink at making sure that this individual is removed as quickly as possible, and then everybody is happy again, right? So this is very much like the situation that you have in your deep tissues and your blood, right? That basically any microbe present in that environment where it should only be your human cells, and our human cells look in many ways quite special on the sort of tree of life. Any microbe that is there is a clear sign that you have a disease and that you need to mount a full-blown immune response, which can actually include quite some collateral damage. And this is in very, very, very sharp contrast to what goes on in your intestine, right? Because in the large intestine of basically all mammals, we have the densest microbial consortium found anywhere on the planet. This will have a few hundred different species in it. They, of course, all look very similar. But in reality, these are massively diverse organisms. So I like this tree of life from 2016. Let's see if I can get myself a laser pointer. Here we go. Where down here you have the opisthosconta, and one of these little green lines is the opisthosconta. And in this little green line, we have really a lot of the diversity we can see, right? Basically everything except plants. So you and squid and mushrooms are all in one little line of this tree of life down here. And the bacteria you have in your intestine span the entire bacterial radiation. So from Gamma-Proteobacteria all the way up to Firmicutes, so the gray bit down here up to the green bit up here. And correspondingly, because these are highly diverse organisms, we also form highly diverse relationships with them. And some of these microbes are important for us. They do things like make vitamin K or help us to extract energy from dietary fiber. On the other hand, in all of us, every single person listening here, if we were to plate out our poo today, you would find opportunistic pathogens, and they're normally in pretty low numbers. They're things like potentially pathogenic E. coli, Klebsiella pneumoniae, Staphylococci, Streptococci, Enterococci. And they're there all the time, but they mostly don't make us sick, right? We just carry them, we keep them at a low level, and this is sort of tolerated by the host. And then, of course, we have many species in the middle that can either be one or the other, or sort of don't do anything useful but don't do anything harmful. And there's been a lot of excitement since kind of the invention of next generation sequencing on how all of this relates to all sorts of different diseases. So you can now kind of pick your favorite disease and type it into PubMed with the word microbiota, and you'll come up with papers where people have gone through patients and controls and sequenced the microbiota and probably found correlations between the disease and the microbiome composition. But the problem with a lot of the data is that we don't really have any good idea about even often whether it's cause or effect. And even where we have some evidence that it's causative, we don't have a lot of information on what the mechanisms are. So I think I have enormously massive respect for the many, many groups around the world who are developing increasingly powerful computational techniques to deal with the large data side of this, right? Looking at all of the microbiome information that is available to us. But nevertheless, even with the best bioinformatics in the world, we have around 1,000 species or up to 1,000 species in each of us. This is going to be more than 2 million different microbial genes. Some of them we can guess at the function via homology, but we're still guessing. And a lot of those genes, we have no idea about the function, right? So there's still a huge amount of dark matter effectively in the microbiome. So there is big progress being made. Things like AlphaFold, of course, hugely powerful in that domain. But nevertheless, our approach has been to say, okay, we're going to work from the other end. We're going to take this bottom-up approach and start with, for example, germ-free mice that have no microbiome at all, or we work with notobiotic species, or we work with things where we can manipulate the genetics very well, things where we have model T-cell receptors. And on top of that, we develop all sorts of interesting technologies to follow this that I'm not going to talk about today, but I would be very happy to talk about later if anybody is interested in analyzing metabolism. Okay, so back to the problem that we were working on when we got into this field. And the main big question we were interested in is what role does the immune system play in controlling this opportunistic pathogen component of the microbiome? And sort of as I alluded to in the first slide with Buckingham Palace and King Charles, most of immunology is focused on what goes on in the deep tissues, where we tend to just think of microbes as being evil and need to be eliminated. And a lot of our understanding of immunology comes from that concept. So there's been a lot of work over decades and decades over what our blood antibodies do. So things like IgG. And these blood antibodies all link to really powerful killing machinery. Okay, so this is kind of the James Bond of the immune system. It will link to things like the complement system, things like phagocytes, things like NK cells that are really, really good at killing pathogens. And that's great if you have an infection in your spleen or somewhere where it's really a serious problem. But we all know, everybody's seen the film, right? You know what happens when all of these violent activities start, that there's always a lot of collateral damage that goes on here. And to a large extent, we want our intestines to keep working, right? Our intestines are not primarily an immune organ, they're primarily a digestive organ. And if you have inflammation in your intestine, you potentially starve. We see this in some of the inflammatory bowel diseases. So what we seem to have evolved in the intestine is a much quieter mechanism of controlling bacteria really at the strain level. And that is secretory antibodies. So secretory antibodies are produced also by B cells, plasma cells, so the same kind of background mechanisms that we use to produce IgG antibodies in the blood, but they're a different isotype. They form dimers, so these Y-shaped molecules are joined together at the tail by something called the J chain. That J chain is recognized by a receptor on epithelial cells called the poly-Ig receptor that transcytoses IgA across the epithelium to the luminal border of the epithelium. And there that receptor actually gets cleaved. So the whole receptor extracellular domain is secreted into the gut lumen, or also into the lungs or into the urogenital tract, together with the IgA. And together that complex is referred to as secretory IgA. It's thought to be really functionally important because this secretory component, so this part of the receptor that is present on IgA out in the gut lumen, is very heavily glycosylated. It seems to play a role in protecting IgA from proteases, which is obviously very useful if you want to function in the digestive system. But beyond that, we sort of didn't have a very good handle on the functions of this antibody. It didn't have any of these so-called classical effector functions that we know from IgG. So it looks more like these little toys that you might have played with as a kid, that it has 4 sticky patches. That's it, right? It doesn't do anything else, just sticks to stuff. And we wanted to find out what that was really good for. You know, if we missed something, is there an effector function that we didn't know about? And so we decided to do this based on really by convenience, by the fact that I was working with Professor Wolf Dietrich Hart at the ETH Zurich, a very, very well established model, which is non-typhoidal salmonella infections in mice. So this is a model organism, right? It's very, very well studied in the lab. The animal models are very reproducible, very easy to work with. We have very big windows to measure things in, but it's also a relevant model organism, right? So salmonella carriage in healthy adults, particularly in farm animals, things like pigs and chickens, transmits then to vulnerable members of the population, so typically the newborns, where it can cause pathology and death. And from animal reservoirs, this can, of course, also transmit into humans. We also have quite a short distance to go from salmonella biology to E. coli biology, and E. coli are really driving a lot of the antimicrobial resistance crisis at the moment. So we think there's actually quite some useful translation that can come out of salmonella work. The first thing that we needed to do when we started this whole journey was to actually be able to induce these secretory IgA responses specific for salmonella. And it turned out when we started out that the main way that people were doing this at the time was to use so-called live attenuated vaccines. So these are based on a specific mutation in the salmonella genome that makes them a little bit less virulent. They don't kill the animals anymore. But nevertheless, it's live bacteria, and those live salmonella bacteria actually for a very long time will colonize the gut draining lymph node tissues, so the mesenteric lymph nodes you can see here, and we have pretty high counts even 3 weeks after vaccinating. They also do cause some inflammation in the intestine, so this is actually pretty strong inflammation at 3 weeks post-vaccination. And those two things together make it very difficult to work out what the role of IgA is in the system, because if you compare your vaccinated mice here, which have loads of salmonella already and they're massively inflamed, to your controls, you're also going to see a lot of effects of things like niche competition and just the inflammatory response on what you're trying to read out. So we devised a method where you can just take your bacterium of interest, you dump it into peracetic acid, which is a very strong oxidizing agent, and you get out a particulate vaccine where the surface structures that we're interested in are effectively unchanged. It obviously doesn't colonize anything because it's dead. It also doesn't induce an inflammatory response because it's dead. But it does induce just as good of a secretory IgA response as the live vaccine. So we had now a good way of introducing secretory IgA against the bacteria we want to target without all these other massive ecosystem changes associated with the live oral vaccines. Okay, we then tested this vaccination model in the murine non-typhoidal salmonellosis model, and this was developed by Wolf Hart's group now about 20 years ago, in fact. There should be an anniversary of this mouse model coming up. So with this model, what you do is you take your normal lab mouse, you treat it with oral high-dose streptomycin. 24 hours later, you introduce a streptomycin-resistant salmonella into this large open niche in the gut, and the salmonella grows to occupy that whole niche. It then expresses virulence mechanisms that allow it to invade into epithelial cells, to induce an inflammatory response, to spread to the draining lymph nodes. And at the point when I moved to ETH, Wolf Hart's group had already set up a system with genetic barcodes in salmonella that allowed you to put some arrows, some numbers on these arrows here. So instead of just being able to count how many bacteria you had in the gut lumen and how many bacteria you had in your lymph nodes, we could ask what the growth rates were here, what the bottlenecks the population was going through were between day 1, day 2, and day 3, how many of the population was seeding the final tissue reservoirs of infection. And there we go, you can have a slide for this. This was combined then with stochastic models to infer growth, death, and clearance rates at each of these stages. And that was work done by Roland Regois at the ETH Zurich together with Claude Leverdo. And the numbers that come out look like this. So around 10% of the inoculum that you give to your mouse can actually start to grow in the gut lumen. And it grows very, very fast during the early stages of infection. So it follows typical sigmoidal growth. It's minimum doubling time is actually about 30 minutes. So it's growing almost as fast in the gut lumen as it would grow in a test tube. Also, during the first day of infection, we have around 300 invasion vents into the lymph node. The bacteria in the tissue are then growing much more slowly, doubling roughly every 8 hours. So we could then look what happens to these numbers when we vaccinate. And perhaps not massively surprisingly, what we see is a massive decrease in the rate of invasion into the tissues. And if we sort of track back to what is it that is decreasing this rate of invasion to the tissues, I've already told you these antibodies are sticky, right? And actually that looks like a pretty good answer, right? So in our naive mice, we have a nice planktonic salmonella population. If this was live microscopy, you'd see these things wildly swimming around. In the vaccinated mice, we've stuck all the salmonella together into these big clumps. There's very few salmonella that are able to still move around in this environment, and therefore they seem to not approach the gut wall. We could also show this with intravital microscopy. And if you sort of do the calculations based on the assumption that only the planktonic bacteria can invade into the intestine, into the intestinal tissues, then the magnitude of the effect that we get by aggregation is more than sufficient to explain this phenotype. So maybe it's not the only thing that the IgA is doing, but for sure it's a big enough effect just aggregating the bacteria together to explain the extent of protection we see by vaccination. So that was great, but we'd set up, this fancy within host population dynamics modeling, and we got a new type of vaccine, and we'd sort of rediscovered classical agglutination, which has been known about since 1896, and that was a little bit disappointing at the time. And then we started thinking about this a bit further, and we had a couple of other bits of data that I will come to in a minute that sort of indicated that there must be something else going on, because this classical agglutinated model, very well established, but somehow it wasn't able to really explain what we were seeing. And it was at that point that we started talking to a very brilliant mathematician called Doug Brumley. And he came from the aquatic microbiology field. And for him, it was very clear, right? It's something that we didn't really think about, because as immunologists, we were always thinking about the properties of the antibodies, and our antibodies need to be able to bind really well in order to cross-link bacteria together. But actually, you can have the stickiest antibody in the world. If you have one bacteria sitting here with me in Zurich and another bacteria sitting with you in Jena, then they are never, ever going to be cross-linked by that antibody, right? You need also the bacteria to encounter each other for the antibody cross-linking to take place. And in fact, in the gut, during early stages of infection, you will swallow maximally about 10 to the 5 salmonella. And in a human, those dilute into several liters of fluid, in a mouse into several milliliters, as well as being diluted into all the rest of the microbiota, right? So it's maybe 10 to the 11 bacteria per gram, and you're introducing in 100,000 pathogens. And so Doug, who is great at understatements, said that this is like bumping into a friend at a music festival while blindfolded, and it's a very, very big music festival, right? You're outnumbered 1 to 1 million. So this formation of these aggregates by clumping during this very early stage of infection, where you really need the protection to work, seems to be not feasible, right? It's just not doable. But there is one case where bacteria that are identical, so 2 salmonella that could be cross-linked by these antibodies, which have 4 identical binding sites, there is a point where those are always in contact, even when you have a low density solution. And that's at the point where the bacteria grow and divide. So when they septate, you have 2 identical bacteria next to each other. And that's a very good time point for IgA to actually be able to cross-link. And we could demonstrate that that is actually what's going on in the gut by doing a very simple experiment where we infected mice with a 1-to-1 mixture of GFP expressing and M. cherry expressing salmonella. And then we looked in the gut content at the early time points post-infection, so about 4 to 8 hours post-infection. And what you see at these early time points is salmonella growing in either green aggregates or red aggregates. And this very clearly, and we did all the statistics on this data, you can only generate these types of aggregates if the bacteria are growing into little colonies that are stuck together by IgA, rather than being randomly agglutinated by IgA. And Okay, this seems, maybe that's seems like a small effect because it's still generating clumps, right? But we could model this process from first principles, and what you can see both from data comparing growing and non-growing salmonella being aggregated in the gut lumen is that as long as our salmonella are growing, we can generate aggregates at much, much lower bacterial densities. So the IgA clumping protective effect can kick in very early during the infectious process. And so it's really a fundamentally different type of protection to classical agglutination. And also, if you kind of drill down into the equations that are behind the lines on this graph here, what we have modeled is the size of this planktonic salmonella population. That would be the bacteria that are able to get into the epithelial cells. And the size of this population is driven by agglutination, removing bacteria, and also bacterial growth, but in the presence of IgA, where some of that growth results in aggregates and a few of the bacteria escape. So this can be broken down into statements that look not quite as pretty as this, but actually are not particularly scary. These statements come from well-established descriptions of bacterial swimming in aqueous environments, as well as turbulent mixing. And the interesting parameters from these equations give us some information about how IgA protection works. So here in C is our concentration of planktonic bacteria. And obviously for both of the terms related to classical agglutination, the relationship here is to the concentration squared, which means that at low concentrations, this is rather inefficient. At high concentrations, it becomes very efficient. This is very strongly concentration dependent, whereas the growth is obviously linearly related to the planktonic population. So what this tells us is agglutination is only efficient at high densities. The other parameter that's interesting here is cell size. So our salmonella are all pretty much the same size, but if you start to think about other pathogens and how agglutination can work, for example, with viruses, here we have cell size cubed, right? So this can be a very, very big effect on turbulent mixing driven classical agglutination. So big cells will always agglutinate better. The size of the cells is irrelevant for the enchained growth, but what is important for the enchained growth is the growth rate, right? So faster growing bacteria will be more efficiently enchained, simply because they have less time to fall apart. The crosslinks have less time to fall apart before the next growth cycle starts. And then obviously the important values here are given as sigma or delta. Actually, delta is sigma -1. And this is just the probability of antibody cross-linking, or the inverse of the probability of antibody cross-linking. And this is related not just to properties of the antibody, but also to the abundance of the antigen that the antibody is targeting, how stable that is on the bacterial membrane, can it be pulled off the bacterial membrane, right? Many other things come into this probability that are not just affinity and avidity. So sort of the overall conclusion is that enchained growth is governed by fundamentally different processes to classical agglutination and reduces the fraction of free infectious bacteria even at very low CFU densities. So this is great. This is was a nice discovery that we could then start to really work with. And one of the first things we could demonstrate with this is that there were two big effects of secretory IgA on horizontal gene transfer in the gut lumen. One of them is that inflammation in the gut lumen is a major driver of lysogenic phage reactivation. So simply because our vaccination prevents intestinal inflammation, we can also prevent lysogenic phage reactivation and reduce gene transfer by generalized transduction. The other thing that's perhaps slightly more surprising is that, of course, this IgA-mediated enchained growth generates population structures. So the aggregates are to some extent clonal. And this actually inhibits conjugation-dependent horizontal gene transfer, because if you have a donor stuck in an aggregate over here and you have an acceptor stuck in an aggregate over here, there's no way that those can undergo conjugative plasmid transfer, and you actually delay massively slow conjugative plasmid transfer in a gut luminal population. So we're having some interesting effects. But then we get down to sort of our favorite topic, which was that we were observing increased clonal extinction of bacteria in the gut lumen when we had a secretory IgA response present. And we were very excited about this, right? Because we thought, you know, everybody's wrong. Everybody's saying this is a boring antibody that just sticks to things. And look, it must be killing stuff because we're seeing extinction. And again, you know, talking with mathematicians and people who know about fluid dynamics, we slowly came to the realization that this excitement was a little bit premature. And actually, if you realize that the gut lumen flows, then we can explain all of this actually still just with our aggregation phenotype. So the nicest way of explaining this to you is to say, okay, this train station here, this is a bit like your gut lumen, right? This is actually Zurich Hauptbahnhof, for anyone who's interested. And you know, these two trains have just arrived, this kind of mass movement of people. So this would be gut microbiota members from the trains on the platform out onto the station concourse. And we're going to put you in charge of a group of kindergarten children, so about 30, let's say, 4-year-olds, who are on this train in platform 4. And your job is to get all of these kindergarten children off this train, through the station, and onto a tram on the other side of Zurich Hauptbahnhof. And if you just take these children and release them into the station from the train, right? They're small children, a bit like bacteria. They tend to do random walks all over the place, at least mine always did. And what you actually expect to happen if you did that with the children is they would sort of diffuse, right? You'd find them pretty evenly distributed over the whole station, and not very many of them on the tram. Not a very successful strategy. And of course, kindergarten teachers have solved this ages ago, right? That what you do is you take your kindergarten children, you get them all to hold hands, and then they will basically be washed along this concourse with the movement of people. Very much like what IgA is doing in your intestine. So by generating these aggregates with clonal structure, we get whole clones washed out pretty efficiently in the gut content. And this we can actually really measure in this clonal extinction. And we could also model this from first principles and again demonstrate that the effect is strong enough to explain what we're seeing without needing to infer any kind of killing mechanisms. Okay, so this seems great at this point, right? We have a nice, safe oral vaccine. It protects from invasion. It protects from inflammation. It's driving this clonal extinction. Why aren't we all taking these vaccines all the time? And there is an Achilles heel in this problem, which is that clonal extinction, which we're inducing with these IgA responses, is of course exactly the same as a selective pressure on these bacteria. And it turns out that the protective antigen on the surface of Salmonella is not these very sort of diverse protein antigens that there are, of course, many of in the outer membrane, but actually the O antigen of lipopolysaccharide. And the reason for this, if you look at these kind of diagrams from textbooks, is not super clear, right? But in reality, the O antigen on Salmonella looks a bit more like the hair on this dog, right? So they look very neat and tidy in diagrams. Actually, this is a complete carpet that covers the surface of Salmonella. And if you're going to grab onto anything on the surface of Salmonella, it's very likely to be the O antigen. The O antigen is a repetitive glycan polymer. It's enzymatically generated. If you pick up a mutation in some of the enzymes that generate this glycan, you can change the nature of the polysaccharide quite dramatically, and you can escape. And particularly when you consider that in the gut, we have huge populations of bacteria, they're growing with maybe 5 generations per day. This is really an ideal situation for very rapid within host evolution. And we set ourselves up basically to lose this game. So we didn't give up at this point. We realized this was going on. We could actually see it in real time. We could pick up the genetic mutations, the genetic modifications that were being selected for by our IgA responses and could map them actually to the O antigen biosynthesis pathway. So what was happening actually was were changes in enzymes that modify the sugar repeats just prior to polymerization into these long glycan chains. There's a good reason for this because this WZX enzyme that flips this tetrasaccharide across the membrane is highly substrate specific. So if you picked up a mutation here, you'd need a matching mutation in this enzyme. Outside, once you get into the periplasm, everything is much less substrate specific. So there's a lot of modifications that can happen at this stage. Interestingly, the modifications are quite small. So they're things like adding or removing an acetyl group from this abaquose sugar, or adding a glucose side chain. But actually, because these things get polymerized, right, ends up being a large change in the charge distribution or in the sort of steric properties of the glycan. And so you really go from having salmonella that looks like this fluffy dog to salmonella that looks like this dog or this dog over here. And the antibodies that bind well to your wild type don't bind very well to the mutants. So what we got then interested in is that, so you can do all sorts of changes here, but there is another step just downstream of this where we polymerize the O antigen. And maybe if we could cut off these types of easy escape routes. So this doesn't come with any fitness cost, changing the O antigen with acetyl groups and glucoses. If we can make antibody responses that recognize not just this original salmonella, but also this relatively low number of alternative O antigen structures as it can produce, maybe we can force salmonella to actually pick up mutations in this WZYB protein that catalyzes polymerization of the O antigen. And I wouldn't be telling this story if the answer was no. So what we could do was construct a vaccine that included all the possible O antigen variants that our strain of salmonella could produce, because you really need enzyme systems to do this. So it doesn't have in the lab, or it doesn't have contact to other bacteria or phage. It doesn't have an infinite number of escape mutations. And if we do that and we challenge the mice that have antibodies against all of these different O antigens, then actually we can isolate salmonella back from the guts of those mice that has lost production of the polymerized O antigen. So looking rather sad, like this little dog down here. That was interesting and academically a good exercise to show we could do that. In reality, out in the real world, completely predicting salmonella evolution and hoping that you've caught all possible evolutionary escape routes is quite bold. So what we realized is that, in fact, what we're doing with these antibody responses is simply altering the outcome of competition between closely related species in the intestine. And that gave us the idea that rather than just waiting for bacterial evolution to generate this less virulent pathogen, we could actually already put in what we're going to call a next generation probiotic that has all the metabolic characteristics. So it competes well with salmonella, but it's not pathogenic. And also it's not recognized by our induced antibody responses. And if we can do that, then we ought to be able to set this up so that our benign competitor always wins the race, right? And this we were actually quite nicely able to demonstrate also in the mouse model, that if we add in a competitor with our vaccine, we can completely prevent intestinal inflammation out already to day 10 post-infection, which is pretty late in the infection process. And if you compare this to a licensed Salmonella typhimurium animal vaccine, it's obviously performing much, much better than this. And if we look now at colonization of the intestine with salmonella, if we have a naive animal in this model, we get colonization out to day 10 of 10 to the 9 to 10 to the 10 salmonella per gram of intestinal content. If we just add in the competitor, we get down to about 10 to the 5 CFU per gram. That's already quite good, but these mice get sick. If we just vaccinate, again, we get down to about 10 to the 5. The animals start to get sick from about day 8, day 9. And if we do this with a combination of the vaccine and the competitor, we can completely eradicate salmonella from the gut lumen. So actually already from day 5 post-infection, we have some animals that have completely cleared. By day 9, day 10, effectively all animals have completely cleared salmonella. They also have no systemic infection and no intestinal inflammation. So with an oral vaccine, we can now actually replace one bacterial strain with another. And we think this is quite a nice system that we can actually use to kind of change the rules of competition in the gut and introduce a strain that we would like to be present, get rid of a strain that we don't want to be present. So for the last few minutes, I want to change topic a little bit and say, okay, we set all this up with salmonella. We're now doing some work with E. coli that's really very similar. What about more diverse species? And we've done a little bit of work with a commensal bacterium called Bacteroides theta omicron. I'm going to call it B theta for the rest of the talk because it's a bit easier to say. It's one of the major dietary fiber digesters in the gut. It had many, many loci for polysaccharide utilization, so it can deal with all sorts of complex polysaccharide polymers. It predominantly grows in the upper large intestine of humans and mice. It's highly abundant in the microbiome. And interestingly for this bug, it produces several different capsular polysaccharides. So it has actually 8 gene clusters for producing these capsular polysaccharides. And the capsule is assumed to be very important for gut colonization of this organism. That said, it's clearly also a metabolic burden on the cell. This here is a picture of Bacteroides. You can see quite how much sugar they're putting out onto their surface. So we can do the same sort of thing with Bacteroides theta. We can vaccinate against it. We do see enchained growth, but it's clearly much less efficient than we're seeing for Salmonella. So I'm not going to talk specifically about immunity or about adaptive immunity to Bacteroides theta for the rest of the talk. I'm going to focus just on its gut colonization potential. So we did the same thing in B theta that we did in Salmonella. We introduced these genetic barcodes, tested everything that it's pretty much neutral. It's obviously slightly harder in these anaerobic bacteria to do the genetic engineering and really be sure that you don't have relevant secondary site mutations coming up during that process. We do see a little bit more wobble in the fitness of these strains than we would have seen with Salmonella, where we can do things like P22 transduction. But we did resequence all of the strains and showed there was nothing super obvious that was affecting fitness in any of them. And we had the huge benefit of collaborating with Eric Martins from Michigan State in the US, who has made a Bacteroides theta strain that has no single gene cluster left to produce any of these capsular polysaccharides. I think he's knocked out something like 128 genes to achieve this. So it's a pretty massive effort. And we were lucky enough to be able to just get this strain and be able to directly put our barcodes into it and study it. So the first thing we wanted to know is how the rest of the microbiome really affects the ability of Bacteroides theta to colonize the gut. So we did some very straightforward experiments where we took mice that had either a notabiotic microbiota, so we have 2 different low complexity microbiotas referred to as LCM, which I'm afraid just stands for low complexity microbiota, or so-called oligo-MM12 mice. That is a microbiome devised by Bebel Stecher in Munich, which contains 12 strains, quite representative of a complete mouse microbiota, also called SPF mice, which have what we were calling a normal experimental mouse microbiota. It may actually be a slightly unusual microbiota, but it's sort of what everybody typically uses around the world. And we did very simple analysis on this data, just assuming a single bottleneck event between the inoculum and growth in the gut lumen, and calculating the probability of any individual strain from our inoculum to actually survive out to our latest time point, which was at the end of day 2 post-colonization. Okay, here's the nice explanation of the different mice we used. And the first thing that we observed was that actually our 2 non-antibiotic microbiota had a very similar colonization kinetics with Bacteroides theta, so roughly the same amount of Bacteroides theta colonizing, and also roughly the same probability of the bacteria to actually colonize. And this was very different with the SPF mice. The SPF mice, we had a smaller total Bacteroides population size and a smaller probability to colonize, but this roughly corresponded, right? So similar order of magnitude change in total population size and the probability suggesting those things are related. Interestingly, if we then went to the acapsular Bacteroides theta, so the Bacteroides lacking this protective sugar coating from its surface, we saw no effect at all on colonization in the notabiotic mice, but we saw a big drop in total colonization efficiency in the SPF mice. So this is going from about 10 to the 8 to about 2 times 10 to the 7, maybe a 5-fold drop. And an even bigger drop in the colonization probability. So actually very few of the clones from our inoculum are actually surviving. They seem to be actually replicating more in the gut of SPF mice. And so this was kind of intriguing, right? That we have a phenotype of needing a capsule that is not so much to do with being in a mouse, but is really due to the context of the microbiota around. So we see 10-fold more clonal extinction in SPF mice. And this was kind of at odds with what what we think capsules are for, right? There's a lot of data on capsules being important for protecting you from the immune system, for example, and the immune system should be in the mouse, not so much in the bugs. So we want to go back and compare to sort of previous experiments that have been done where wild type and acapsular strains were actually competitively colonized into mice. And when we did that, we saw actually, yes, the acapsular strain does have a fitness defect everywhere compared to the wild type. So in competitive colonizations, even in germ-free mice, it's about 10-fold less fit than the wild type, going out to being around 10 to the 5-fold less fit in our SPF mice. And correspondingly, we see this change in probability of colonization. So this then raised the question of why we see equal colonization probability for acapsular strains in single colonizations, particularly in things like these gnotobiotic mice, but we see a reduced colonization probability in competition. And it was then that we noticed that the acapsular strain was actually growing with a considerably longer lag phase, also in vitro, but particularly in the mice. So here in this panel E, these are individually colonized animals. So one group of mice in blue, a second group of mice in pink. And if we just take feces over time and look at how much Bacteroides is there, even with no competitor at all, the acapsular strain is growing up clearly more slowly. There's a longer phase, but it's reaching similar levels of colonization by the 48-hour time point. So if we were to just look here, it would look like not a very big effect. If we're looking at 24 hours, we have 10 to the 5, 10 to the 6 bacteria in the wild type, and only about 10 to the 3 bacteria in the acapsula. And this kind of corresponds to if we do competitive colonizations, actually the wild type has largely filled the niche for acapsular B theta already by about around 20 hours post-colonization. So you can't really have any further expansion of the acapsular B theta after this point because its competitive niche has been already occupied. So actually this brought up something we hadn't really been thinking about, which relates to lag phase and intestinal clearance. And we went back to our mice and did some very basic experiments where we just collected feces over 24 hours and calculated how much dry mass of feces is lost by a mouse over a day. And this is about twice as much in germ-free mice as it is in SPF mice. We then took fresh cecum content from germ-free and SPF mice. We calculated the total weight P and post-drying and also the water content. And again, what you find there is that in germ-free mice, we have a higher water content than in SPF, but actually not that much dramatically higher. It's about 80% water compared to 76% water. But the cecum volume is much, much higher in germ-free mice. So we have around 2.5 grams of cecum content compared to about 0.6 grams of cecum content. We make an assumption of minimal loss of dry mass during colon transit, which is probably not extremely unfair. And if you use that data, you can go back and calculate the amount of cecum content that is lost during a normal day in a mouse that accounts for the feces, and therefore the dilution factor of the mouse cecum content per hour. And in germ-free mice, this turns out to be about 0.12. I mean, the SPF is about 0.23. So we have a faster dilution rate in colonized mouse than in non-colonized mice. And this becomes then highly relevant if you have a lag phase, right? Because during your lag phase, you're not growing at all, but you're being continuously washed out by this dilution. And so we were able to actually include this lag phase into our models of how B theta is colonizing. And when you include both the slightly lower growth rate for, which is really a slightly higher clearance rate for the acapsular B theta, and then also this lag phase, we can get relatively good coherence between our estimates from the model shown in green and actually our data. This is looking at the ratio, the expected ratio of wild type to knockout B theta in the mice. That said, it is a relatively small effect compared to intestinal inflammation. So we do see some effects of the microbiome composition on the ability of B theta to colonize. If we instead do an experiment where we pre-colonize the mice with Bacteroides theta and then we use salmonella to induce a strong inflammatory response in the gut, you can actually cause a very, very dramatic population collapse of the Bacteroides theta population. So more than 100, more than one in, or less than 1 in 100 of these bacteria survive the induction of inflammation in the gut. So this was also quite informative as to how much of an effect inflammatory responses in the intestine really have on the gut microbiota. Okay, so just to sort of conclude, how can the immune system control the microbiota? Intestinal antibodies can exert a strong selective pressure via aggregation. We can manipulate via oral vaccination, particularly things like oral live whole cell inactivated oral vaccines are good for things like salmonella. If we want to induce anti-capsule responses, we need oral glycoconjugate vaccines, which is another big research topic in the lab. But if we can get the right IgA responses, we can really manipulate selective pressures in the gut. We can use this to replace an unwanted strain with one that has beneficial characteristics. And we're hoping we can expand this to more diverse strains than salmonella and E. coli. But proviso to this is that we need very detailed knowledge of the cell surface antigens that need to be targeted by IgA, with a proviso that these are really most often glycans, which are unfortunately a bit of a pain to work with. It should also be noted that the IgA is really one of many selective pressures in the intestine. So obviously with the Bacteroides theta work, I showed the real effects there are coming from things like nutrient availability and flow of intestinal content, inflammatory mediators and bile acids, for example. But with a more detailed fundamental knowledge of how these systems work, we can then much better start to translate this and think of how we can actually use this type of systems to get reliable therapies in the gut. So I want to acknowledge all of the people who've contributed to this work. I think I've mentioned many of them on the way through, also all of our funding bodies, my group, so the people involved in this, particularly Daniel Hosses, who did all of the Bacteroides theta work, Katrin Moore and Verena Lench did a lot of the Salmonella IgA work, supported by Salma Aslani, Georgia Greta, Marcus Arnoldini, and Tim Keyes. And with that, I thank you very much for listening, and I look forward to your questions.
Thank you very much, Emma, for a really nice talk. Does anybody have any questions? I have some questions.
Yes, I can see the chat. So I can't
maybe
get the...
I'll start.
Maybe I can open this. There we go.
So I thought this thing that you said towards the end about the lag phase was really interesting. So the acapsular strain has a long lag phase, but do you understand why?
No, it's a very easy answer. No, we don't understand why. We have some hypotheses, so which we sort of tried to test, but the results were always quite messy. So I'm not confident in concluding anything from them. So one would be that because they lack a capsule, they struggle